We will continue from there. Okay. Now, here is a question from a 22 exam. Explain why this treatment does not prevent people becoming ill if they are infected again with the same pathogen. So they're talking about antibodies can be collected from human blood donors and used to treat people that may have been infected with the pathogen. This prevents them becoming ill with the disease. When I take an antibody from one organism and I inject it in another organism, this is which type of immunity, Noor? Noor? Hello?
Yes, Miss. Yes. Sorry, I'm lagging. Can you ask the question again?
If you took antibodies from one organism and you will put it in the body of another organism to treat an emergency situation, what is this? So
Artificial.
your body did not make the antibodies. The antibodies were taken from another organism, ready-made antibodies from another organism. This is which type of immunity?
Artificial.
Farouk, can you tell me? Come on, boys, your exam is tomorrow.
Artificial,
Okay, so wake up with me. When you take ready-made antibodies, this is
artificial
active
passive,
immunity or
Artificial,
passive?
Miss. Artificial.
passive, artificial passive immunity.
Passive. Yes.
Can you hear me, everyone?
Yes, we can.
Yes.
Okay, the problem is, I don't know what's wrong with the connection today. Okay, back to the question. You can see my screen? This question is asking you about antibodies that have been taken from one organism and they are injected in the body of another organism to treat an emergency situation. So this type of immunity is which type? Hello?
Artificial passive immunity.
Of
Artificial
course.
passive.
Yes, so this is the first thing you will mention. This type of immunity is artificial passive immunity. Now, explain why this treatment, this artificial passive immunity, does not prevent people becoming ill if they are infected again with the same pathogen.
Because it provides short-term immunity. There is no
Yes,
memory cells produced, and there is no
yes.
antibodies produced,
No
and
antibodies, no memory cells. Very good.
the immune
Okay,
system is not stimulated
exactly. The immune system is not activated. Now, tell me, those antibodies that have been injected, where do they go?
to the blood.
Yes, and then do they remain in the blood forever or they're broken down?
They're broken down.
Yes, they will be broken down after some
Broken down.
time. Yes. So this is artificial passive immunity. Now let's exploit the chance and revise active immunity. Talk to me about active immunity and what's the difference between active and passive.
Active means long-term immunity.
Yeah,
Memory cells are produced and antibodies are produced,
yeah,
and the
correct.
immune system
Memory
is
cells and the immune system is activated. But this is so summarized. What is injected in active immunity if you are talking about a vaccine?
a dead or weakened pathogen or an antigen or a foreign antigen.
Good, good. And when this dead antigen or weakened antigen is injected into the body, what happens inside the body?
Uh, the macrophage engulfs it
Okay.
by phagocytosis,
Yes.
and then forms a phagosome around it. Then the
Mhm.
lysosome forms with the phagosome, forming a phagolysosome and releasing hydrolytic enzymes such as ligase and nuclease to break down
Okay,
and
you
kill the
explained
pathogen.
that. Okay.
And then
You explained phagocytosis in detail. And then,
the macrophage turns into an antigen presenting cell
okay.
and then finds a complementary T helper cell by clonal selection, that they undergo clonal expansion, and then
Wait,
the
wait.
T
The T helper cell is going to become activated and it will release what?
cytokines to stimulate
Yes.
the T lymphocytes and the T killer cells. The
Wait,
T killer, yeah,
Farouk, stop. They will activate the B lymphocytes and the T lymphocytes in general. And
yeah,
then the B lymphocyte is going to go through colonal selection, colonal expansion, and it will differentiate. Please understand the order. First, the immune system is activated, which is what you just said. The immune system is activated. In this activation, you will have the entrance of the antigen and then the phagocytosis and antigen presentation, etc., etc. And also within the activation of the immune system, it involves colonal selection and colonal expansion. What I noticed is that you're getting confused. Both the lymphocytes, both the B and the T lymphocytes, go through colonal selection and colonal expansion. We have a foreign antigen here. I want to know who's going to attack it. Let's say one of the B lymphocytes will do the job. We have many B lymphocytes, then we will select one of them. This is colonal selection, that the B lymphocyte with the complementary receptor will be selected to kill the antigen. After the selection is done by the body, it will go through colonal expansion and it will start multiplying. And then it will differentiate. B lymphocytes actually differentiate into one type of cell only. What is it? What is it, Noor? That produces antibodies.
What produces the
Maryam.
antibodies?
So the B lymphocyte differentiates into what type of cell that produces antibodies? You know it. It starts with a P.
Uh,
The
the
cell that we extract when we are doing monoclonal antibodies using the hybridoma method. Come on. Plasma?
plasma cells.
Yes, you know it. You just forgot. The plasma cells. And then these plasma cells will produce antibodies. Now, what about the T lymphocytes? The T lymphocytes, if they are selected, then this is colonal selection. And then they will divide by mitosis to increase in number. This is colonal expansion. And then they differentiate. T lymphocytes has more than one type. It will differentiate into more T helper, into T cytotoxic, or T suppressor. T suppressor is not important now because we are still in the infective stage of the disease. T cytotoxic or the T killer does not produce antibodies. T killer does not produce antibodies. Okay, I always see this in the answer. T killer cells that produce antibodies. No, it's only the plasma cells that produce antibodies. T killer cells, they're called killer. They directly kill the infected cell. Okay, they have several methods. One of the methods is that they create holes in the cell wall of the pathogen, if it does have a cell wall, by secreting hydrogen peroxide or perforin, and it kills the pathogen. And at the end, after all the killing is done and we're finished, these cells will change into memory cells. So the plasma cells, after they release the antibodies and the antibodies do their job, the plasma cells will become memory cells for a secondary reinfection to provide immunity. And the T killer cells, after they do their job, they will also convert to memory T cells. So in case this pathogen enters the body in the future. Okay. Now, Noor, no, Faru, everything you said is correct, but you explained phagocytosis in general. Mention all those steps when you are asked directly about phagocytosis. When you are asked about the reaction of the body to a pathogen, list everything, including phagocytosis, but not in all this detail, or you will not have space. You will mention the entrance of the pathogen, the activation of the immune system, phagocytosis, outline it, colonal selection, colonal expansion, differentiation. And you will discuss both B lymphocytes and T lymphocytes. Are you with me, everyone? And of course, mention that this will provide long-term immunity, but it's not immediate acting. This takes time. This primary immune response takes time. That's why the person is sick before he is better. Are you all with me? But in this question, because we are talking about passive immunity, none of this happens. So if you are infected with the same antigen, with the same pathogen in the future, you will have to take another monoclonal antibody. Did you all get it? Any questions?
no.
Okay, good. Now, this is a yes, the graph about enzymes. Now, I will not waste time reading the question. Look at the graph and tell me, here is the normal graph with no enzyme. Here is the graph with an inhibitor. Now, the graph obviously reaches to the same Vmax. Now, this is which type of inhibitor, competitive or non-competitive? Who will remind me? I would don't have to say names. Shout the answer if you know it.
Competitive
Mariam, you're there?
Competitive.
Yes. Competitive inhibitor. Let's remember together the features of a competitive inhibitor. What does it do to the initial reaction rate?
Decreases.
It decreases. So it decreases the initial reaction rate. What does it do to the Vmax?
Stays the same.
It stays the same. Very good. We will eventually reach the Vmax. Maybe it will take more time, but we will eventually reach the same Vmax. What does it do to the Michaelis constant? Increases or decreases?
It increases.
Correct? Because why? So the Michaelis constant increases. Why? What does the competitive inhibitor do to the affinity of the enzyme?
The affinity decreases.
Yes. Very good. Perfect. Now let's exploit the chance and revise the non-competitive inhibitor. The non-competitive inhibitor, if you have an original graph and you are requested to draw the graph or sketch the graph of a non-competitive inhibitor, how will you draw it? Above, below, describe.
Below, and it does not reach the same Vmax.
Perfect. Okay, it does not reach. I added here that for you. Okay, now tell me, how does the non-competitive inhibitor affect the initial reaction rate?
It decreases.
It decreases. How does it affect the Vmax?
It decreases also.
Decreases also. How does it affect, where is it? Didn't I how does it affect the Michaelis constant here? Hmm?
It stays the same.
It stays the same. Perfect, because it does not affect the affinity. Now, here I was answering this revision sheet for you, and I exploited the chance to revise competitive and non-competitive inhibitor. Okay, and this graph, it's easy. As long as you understand it, it will be very easy. When you come to talk about the competitive inhibitor, memorize this sentence that as the substrate concentration increases, there is an increased chance. No, 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 not this one. Where? Mm, yeah. An increase in the substrate concentration cancels out the effect of the competitive inhibitor. That is why the Vmax will eventually be reached. When they ask you, explain why does the Vmax, how come the Vmax does not change with competitive inhibitor? You will mention this sentence: as the substrate concentration increases, it cancels out the effect of the competitive inhibitor, but not that is not the case with the non-competitive inhibitor. Can you tell me why? When you increase the substrate concentration, does it cancel out the effect of the non-competitive inhibitor?
No,
No, very good. Why?
because the non-competitive inhibitor binds with the alternative site, which causes the shape of the active site to change. Therefore, an increase in substrate will still not be able to bind with the active site and form an enzyme substrate complex.
Perfect. Okay, so now we revised all this part. Here so acts as an inhibitor of the enzyme. Here you will, in this question, you will say it's competitive because the Vmax is reached and all the features that we said, we revised all. Suggest why large outbreaks of cholera occurred in countries listed in table 5.1, but there were very few cases in most other countries. Let's exploit the chance and revise cholera. It's the most common disease that they asked for, cholera and HIV. So while you're studying, please focus on those two. The other two diseases or the other diseases, they're not very common. Cholera, TB, and HIV. So let's exploit the chance. What is the pathogen that causes cholera?
Vibrio cholerae.
Quickly. Yes, Vibrio cholera. It's virus, bacteria. What is it? Protoctist.
Bacteria.
What? It's a bacteria. Okay, now the mode of transmission. Mode of transmission.
Fecal
How is it?
oral.
Very good. Oral fecal route is a very nice key term. Now this needs a little bit of elaboration. What does oral fecal route mean?
That it's transmitted from feces to the mouth,
Yes.
to the food that a person
Yes,
eats.
you're
So
Very good. Oral fecal route is a scientific term, meaning that the pathogen is always in the feces. So the diarrhea that comes out of the patients is very, very dangerous if it infects the waters and you drink this water, you use it in washing, cleaning, cooking. There will be an outbreak of cholera. Always in wars or in natural disasters, cholera is the first disease that becomes an epidemic. Okay, so this is the mode of transmission. Now, how does the cholera cause diarrhea? This is O level, by the way, but I see questions asking about the sequence. How does the cholera cause diarrhea?
The bacteria causes irritation at the guts, which will cause
Yes,
chloride ions to be released inside the guts, and the release of chloride ions will cause a decrease in the water potential of the guts. Therefore, water moves from the capillaries, and the feces will become watery and move fast. Therefore, it takes more time to absorb the water back, and this causes diarrhea.
excellent. Okay, I hope you all remember that from O level because they can ask you. And I think it's mentioned also somewhere in your notes. Good. Now, treatment. Noor, tell me, how do we treat cholera? Quickly.
By oral rehydration therapy
And
and prevent يعني
it's
don't
caused
drink
by a bacteria. Yes, these are preventative measures, but this is caused by a bacteria, so we can treat it, use it. Come on.
an
Antibiotic.
antibiotic, yes,
Yes, there is a vaccine for cholera, but obviously it's not very successful. Why not?
because the antigen of the bacteria is constantly changing.
Yes, because of there's antigen variation, antigen concealment, okay, because it mutates, because it's not stable. Now let's go back and answer the question. We were revising cholera, but we did not answer the question. Why outbreaks of cholera can spread in particular in some countries, but not in other countries?
Countries with poor يعني
Mm.
countries where regular contaminated water with sewage يعني a country where مثلا a natural war is going ولا
Yes. Yes, when you see a question like this and you have 5 marks for that question, you need to mention 5 points. Any contamination of water, okay, so the water is contaminated. There is no proper sanitation, no proper sewage system. Sewage is used for the irrigation of crops, or sewage is contaminating the water. There's a natural disaster, there's a war, no funding,
no vaccines,
no vaccines. Very good. You just mentioned
expensive to
5 points,
store.
okay? Excuse me, expensive. Yes, everything we discussed it yesterday, but Farouk was there. Describe the role of the transfer RNA shown in figure 6.2. Okay, why did I put this question? Because I want to revise the steps of protein synthesis. This question asks about translation, but let's revise it from the very beginning. Transcription and translation. Who will remind me? What happens in protein synthesis? What are the two main steps? Quickly.
Transcription and translation.
Yes. Okay. Now, transcription, and there is post-transcriptional modification, and there is translation and post-translational modification. Don't forget those parts. So let me write it down. Transcription and post-transcriptional modification. And we have translation and post-translational modification. Now, what happens in transcription?
The
Your
the
exam is
mRNA
tomorrow. You know
is
that you
formed.
have to throw the answer. Excuse me?
The mRNA is formed when with the DNA helicase breaks the bond between a part of the DNA, forming the template strand. Free RNA nucleotides arrive at the template strand, and they're complementary. And the RNA polymerase enzyme forms the sugar phosphate backbone of the RNA, forming the mRNA molecule that leaves the part, and then the DNA starts to coil back.
Very good. This is transcription. Perfect. Did you all understand what Mariam said? She mentioned the name of the enzyme and everything. Now, what about the post-transcriptional modification? Noor, can you explain after the messenger RNA? is made, and it still did not leave the nucleus. It's still inside the nucleus. Number one, it's called the primary transcript or the primary messenger RNA. Or what happens to this primary transcript? Yes, Noor, are you with me?
Yes, Miss.
What
I'm
happens
lagging.
after we copy? Okay, after we copied part of the DNA, like Mariam said exactly. We have the primary transcript or the primary messenger RNA. What is the modification that happens here? Farouk, do you remember?
The primary transcript goes to the ribosome.
Before it goes to the ribosome, boys, something happens to the messenger
Oh,
RNA before I will not directly translate.
It's checked the ones the introns are splicing.
Yes. Splicing. Yes, my dear, there is no chance here for confusion. I said, Noor, study well. Well means you will answer with confidence. You will stay, you will not spend any time thinking. Now, what is splicing?
you remove the non-coding introns and join the coding exons.
Excellent. We remove the introns because they're non-coding, and we fuse the exons. Now, there was a question that said, suggest what is the function of the introns? They don't really know the function of the introns. They're not really sure what the introns are for. The introns, what they know that they're non-coding, but if you are asked to suggest these introns, what is their function? What suggestion can you give? Tell me. I will tell you, even though you will not leave the question empty. The introns always, I saw this question 2 times in 22, 23 in the last years, and the same suggestion was in the mark scheme, which is they may have a role in gene expression. So please remember that. They can be involved in expression of the genes, which genes will be transcribed and which genes will not be transcribed. Didn't you take in A2 the promoter?
Yes.
Yes. So they can have the same function as the promoter, but they're not sure. So if they ask you, suggest a function of the introns, you know some facts. Actually, you only know one fact, that the intron is non-coding, it is removed, it is not part of the translation process, it is not part of the final polypeptide chain. And there was a question that I answered yesterday with Farouk, the comparison between the exons and the introns. So you know some facts. But suggesting what the function of the introns is, it can play a role in gene regulation or gene expression. Please don't forget that. Gene regulation or gene expression. Those that are in A2 will understand more about that because there are some parts of the DNA that are binding sites for the messenger RNA. They're called the promoters, and the intros can act as a promoter. Okay, so in case they ask you suggest functions of the intron, it can play a role in gene expression or gene regulation, controlling which genes will be transcribed and which will not be transcribed, if we will make it nice and clear. Okay, hope this is clear. Now, translation. Farouk, tell me what happens in translation, which is actually the question.
The tRNA provides
Yeah.
the anticodons, complementary anticodons,
Yes.
and is connected to an amino acid.
Okay.
When the ribosome can only have two tRNAs connected at the same time.
Yes, of course.
And when
And when does the translation stop?
when I stop coding.
Yes. Okay, so the transfer RNA will bind to the ribosome, and the transfer RNA does not have the amino acid attached to it. It's not ready. It will pick the specific amino acid, okay? And it will bind to this amino acid binding site. This happens in the cytoplasm. This is the anticodon that is complementary to the codon on the messenger RNA. How does the anticodon and the codon fuse together? What are the bonds that are made to fuse the bases together?
The amino acids.
No, what are the bonds that are made between any base, any complementary bases?
Phosphodiester.
My dear, no.
It's right here, hydrogen.
Hydrogen bonds. Okay, so the hydrogen bonds are the bonds that are made between any complementary bases, and no enzyme is involved here. Okay, this is a simultaneous action. The phosphodiester bonds happens in the sugar phosphate backbone, whether in the DNA or RNA. And the transfer RNA has an amino acid binding site, and of course, they are specific. Every transfer RNA binds with a specific amino acid. Two amino acids are brought close together, a peptide bond is made. The first amino acid it leaves because we only can have two transfer RNA molecules at any ribosome at any time. And the translation will continue until we stop. Okay, this is translation. Now, I told you that there is a post-translational modification step. What happens in the post-translational modification? We have here, at the end of translation, the sequence of amino acids. What structure of protein is that? Primary, secondary, or tertiary? Don't know why my questions are not heard. Hello? Okay. This sequence of amino acids, it's which level in protein structure? Primary, secondary, tertiary, or what? Can you someone give me an answer?
Primary.
Primary, okay, but this is not the final structure. Then modification, this is not the end of the protein making. What happens in the post-translational modification? Where does it go so that more modification takes place? Yes, to the Golgi body, to the rough endoplasmic reticulum. In these two organelles, more modification takes place. Now discuss or give me examples of the modification. What do we do in order to convert? What did you say?
You remove the first amino acid or fold it or like
Exactly. Perfect. We remove the first amino acid because it doesn't have really any function. It just says start and end. So we remove the first, we remove the final amino acid, and then the remaining strand, we will fold it into a secondary structure or a primary structure. We can add a carbohydrate chain to make it a glycoprotein, etc., so that it's functional. So this primary chain of amino acids is not still the last step in protein synthesis. There is a post-translational modification. It will go to the Golgi body and rough endoplasmic reticulum. Some modifications will take place. To give examples, I'll add carbohydrate. I will add a lipid chain. I'm going to remove one amino acid. I will remove the start and the ending amino acids. I will fold them so that it has a precise three-dimensional shape. I can add more than one polypeptide chain together so that it becomes a quaternary protein. And this will be the final product. Good. Okay. Outline the role of the co-transporter proteins in companion cells. Now, I wanted to revise that. We revised it yesterday, Farouk. Yes? Can someone tell me, Noor, can you tell me what does the transporter proteins, they do exactly in companion cells?
The co-transporter proteins allow sucrose to enter the cell by secondary active transport.
You are correct. Can you tell me what starts first?
Hydrogen.
Like what helps?
Hydrogen pumps hydrogen or protons out of the cell,
Correct.
which creates a proton gradient.
Excellent. And there's
And
another nice name that they like it, electrochemical gradient.
okay, electrochemical
Okay.
gradient.
This is the proton
Um,
gradient. When it moves down, it will move down. Okay. Huh?
Then
And then?
the protons re-enter the cell with the sucrose by secondary active
Yes.
transport.
Okay, now I expect that you solved a lot of questions. And if you notice, they always say accept an annotated diagram, accept a diagram. So in most of the questions in paper two, you can add a small diagram to the side of the answer, and this will actually give you a grade. It's accepted and it is preferred. So if you are coming to talk about this part, why don't you draw a small diagram? Let me erase it and draw it with you. It's very easy because it helps you remember and it looks very elegant and sophisticated and it is accepted. So you will draw, for example, if they're asking you about the movement of the sucrose into the companion cell in plants, because this is the example that we took. This is the cell membrane. This is the cell wall. This is the cytoplasm. Okay. And you will draw the two proteins. The first one is the hydrogen pump. And the second one is the co-transporter. Look at how I will draw it. It's very easy. It looks like a W. Look, 1, 2, 1, 2. This is the co-transporter protein. And don't forget to label. Okay. So here you will mention that the hydrogen will be pumped against the concentration gradient until you have a proton gradient. Just like Noor said. And then you will take, I change color, but you can do that. And then it will move down a concentration gradient by facilitated diffusion down an electrochemical gradient. And as the hydrogen is pumped, as the hydrogen moves down a concentration gradient, the sucrose is going to move against a concentration gradient by secondary active transport. A diagram like this gives you a mark or two. It's very easy. Okay. Also, use those diagrams while you're studying because they will remind you of everything and in proper order. Good. Now, this question was in the 2022 exam, and I really wanted to revise this with you. Which one, A or B, is taken by a transmission electron microscope and which by a scanning electron microscope, and why?
B is the transmission electron microscope, and A is
Good.
the scanning, because
Yes.
A has the look of the outer surface of the cell or the subject, or yeah, and the second one, it has a 2D shape, or the inner parts of a cell can be seen, and there is a small field of view in the transmission electron microscope, and in the scanning there is a higher or a larger field of view.
Yes. Perfect. Excellent. Okay. In the lesson cell structure, please focus on the two different types of microscope, the question and the magnification equation, of course. And what do they also ask about? Usually they stress on this part, the microscope part. And I think I sent you a comparison between both. It was not in a table, so I put it in a table. Look at this because I'm happy to revise this with you. Everything Maryam said is in this nice table here. I will add it next year to the notes, so I thought you should have it. The transmission from the name, the electron beam will go inside the specimen. I cannot here, unfortunately, do any markings because it's password protected. Scanning electron microscope from the name, the electron beams will bounce off the surface in case you are asked about the application. Now, the size, the transmission electron microscope, like Maryam said, a smaller field of view, smaller, and the samples are very thin. Scanning electron microscope, you see a bigger field of view. And the transmission electron microscope is a two-dimensional image. The scanning electron microscope, three-dimensional image. Look at the picture here. Where is it? I'll show it back. Where did it go? Oh, here. This is a two-dimensional. I only see two sides. And this is three-dimensional. I can see the top. I can see like this is three-dimensional. Good. Fine. So the magnification and resolution, as long as you can see a smaller and a bigger field of view, then the transmission electron microscope has a higher magnification and resolution, while the scanning has a smaller magnification and resolution. And please study magnification and resolution. How do we calculate the resolution? Who will remind me?
It's half the wavelength
Yes,
of the wave.
it's half the wavelength. Okay, and the higher the resolution, the more. What does the higher resolution mean?
The
Less wavelength.
No, not the less wavelength. The resolution is half the wavelength. So the smaller the wavelength, the higher the resolution. They're inversely proportional. And a microscope with a high resolution, this means that you can see the distance between 2 points. Even if they're very close, you can see the distance between the 2 points. If the resolution was low, then you will see those 2 points as 1 point. And if the size of the specimen is smaller than the resolution of the microscope, then you will not even see it. Are you with me? Do we revise this part?
No, it's easy.
Hello? Sure? Like,
Yes.
let's say the resolution of the light microscope is 200 nanometers. If the object is smaller than 200 nanometers, will you be able to see it?
No.
No, I will not be able to see it. Why? Does it interfere or it does not interfere with the light waves?
Doesn't.
It doesn't. Very good. Go back and please study this part. Don't ignore it. And especially before paper 3. Here you have light waves. Good. Now, if you have a very small object like a ribosome that does not interfere with the light wave, so the light wave will pass straight through it and you will not see it. But if you have a big object, this object will interfere with the direction of the light waves and the light waves will change direction. You will be able to see it. Okay, so the resolution is half the wavelength. The wavelength, of course, you know very well, is the distance between two successive crests or troughs. Half the wavelength is equal to the resolution. So they are inversely proportional. The higher the resolution, the more you can see better with a microscope, the more detail and the distance between 2 points. If the object is below the resolution and size of the microscope, you will not even be able to see the object because it does not interfere with the waves of the object, of the microscope. Okay? The object is too small. Do you remember this part or you don't?
Yes.
Okay. State and explain the importance of mitosis by stem cells in a small intestine. Now, a chance to revise. What is stem cells? What are they? Quickly, 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 quickly. What's the definition of stem cells?
Stem cells are
Yeah.
unspecialized cells that divide by mitosis to produce identical daughter cells that can be specialized.
Yes, very good. They divide by mitosis to give you genetically identical cells, more stem cells, or they can differentiate. Now, why are stem cells present in some organs? What is their function? Noor, can you tell me? When they can divide by mitosis and produce genetically identical cells, why are they important?
To replace damaged cells.
Very good. You are right. You will mention the functions of mitosis. You will replace damaged cells. You will repair. When cells are replaced by genetically identical cells, you will repair the whole tissue. Yes? A very
Yes.
easy question, by the way. You will repair tissues, you will replace cells, you will have to produce cells that are genetically identical. That's why we start with stem cells. Okay? So what happens is that we have a stem cell. This stem cell will divide by mitosis to give me more stem cells that I will use in the future. And then some of them can differentiate. Let's say we lost a skin cell. So one of them will differentiate into a skin cell. Now this skin cell can continue dividing by mitosis to produce more skin cells, more skin cells, more skin cells to replace those cells that have died. State two differences between sucrose and fructose other than the number of carbon and hydrogen and oxygen atoms present. Now let's revise together biological molecules. What is a sucrose and what is a fructose? Silence. Which one is a disaccharide? My goodness. Of course, it's a disaccharide and it's made up of which monomers?
Alpha glucose and fructose.
Very good, alpha glucose and fructose. What is the bond here?
1 to 4 glycosidic bond.
No, 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 no. This is in starch. This is in maltose.
1 to 2.
1 to 2 alpha, 1 to 2 glycosidic bond. Mariam, let's draw it together and remember it. You will draw one alpha glucose, my dear. Here, 1, 2, 3, 4, 5, 6. Let me change color. Oxygen, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. 6 is outside the ring. And then I go to 2 and the hydroxide or the hydroxyl groups are alternating down, up, down. This is alpha. So I'll go to carbon 1 and the hydrogen is above and the OH is below. Now the fructose. We will study the inverted form of the fructose. 1, pay attention. 2, 3, 4, 5. Okay, we have an O here, and the carbon as in an anti-clockwise direction. We have carbon 1 up there, 2, 3, 4, 5, and 6. CH2OH. This is carbon 1. CH2OH. This is carbon 6. And then you go to carbon 2, down, down, up, up. Okay? Are you with me? Did you forget it? And then the bond
No, I just
here.
got confused.
Okay. H2OH. We'll take one hydrogen and leave the oxygen here. So this is the condensation reaction and water goes out. Look, the bond here between carbon 1. Let me highlight it. Between carbon 1 and carbon 2. Okay? This is our alpha 1 to 2 glycosidic bond. Good. So what is the fructose? It is a disaccharide or a monosaccharide?
Monosaccharide.
Correct. Does it have any bonding?
No.
No bonding. Very good. Okay, which one of them is reducing and which one of them is a non-reducing sugar? Can you tell me, Farouk? Which is a non-reducing sugar? Actually, it's the only example that you studied of a non-reducing sugar. Sucrose or fructose, Farouk?
The sucrose is reducing.
I did not hear you, my dear.
The sucrose is the reducing sugar.
Are you sure? So the sucrose will give me a positive result with Benedict?
No,
Hmm.
it's not.
Okay, so the sucrose is non-reducing, my dear. It does not donate electrons. It does not reduce the Benedict, and it will give me a negative result with Benedict. It's the fructose that is reducing. Reducing means they donate electrons, and this electron will be taken by the Benedict, and it will give me a positive result with Benedict. Okay, don't forget those details. Good. Now here, let's remember together the triglyceride. What is this? And what are these? And what is the bond? Who will tell me what is A?
It's a glycerol.
Correct. It's a glycerol. A glycerol. Okay, we have 3 carbon, 2, 3 hydroxyl group. It's very easy. A simple alcohol and H, 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 H. Good. And what is this bond? B.
Ester.
Ester. Well done. And what are these?
Fatty acid.
Very good. Hydrocarbon chains. Now, they will ask you in the common question, what's the difference between this fatty acid and this one?
The
And those
palmitate
two.
is saturated.
Yes, perfect.
The palmitate
What
is
does saturated mean?
there's no carbon double bonds. It's the
Yes,
maximum
you are right. Maximum amount of
hydrogen.
hydrogen. Well done. Here, these are unsaturated. Good. There are double bonds. If we have only one double bond, it's mono unsaturated. More than one double bond, poly unsaturated, means those bonds can break and accept more hydrogen. So we just mentioned that. Okay. Outline the similarities and differences between the introns and the exons. Didn't we just answer this question?
Yes.
Yeah, we answered it.
We answered these questions, and I don't want you to forget that in case they ask you, suggest a function of the intron, they can have a role in gene expression or gene regulation. Okay, and please don't ignore the mark scheme. You know why? Because I added some things to the mark scheme from the notes just to remind you of things. Like here are the introns and the exons. I really liked this difference. Okay, look at it. Like it's a form of studying. Good. Okay, now this is important. We answered that the last time, or the last exam we answered was this one. What is T? What is T? This is the section through your respiratory system. T is what?
Ciliated epithelium.
Yes, the ciliated epithelium. Now this lesson, gas exchange, they ask a lot about the gas exchange, so please study. the ciliated epithelium. And in the last class or the class before the last, we discussed what we will write if you are asked about the description of the ciliated epithelium. Mariam, can you remind me?
Yes, the ciliated epithelium cells are columnar
Yes.
with a basal nucleus,
Yes.
and they have goblet cells that
Very
and
good.
milk glands that release mucus that traps the pathogens and dust particles. And the
Now, what
cells
is
are present on a basement membrane.
Yes. Okay, now this is for all of you. I saw this question repeated in the last, more than once in the last years, and they want you to describe the ciliated epithelium and the cartilage. Okay. And again, I added that to next year's notes. I added this for you so that you have everything. You have this picture in your notes, and I added one sentence. I cannot mark it here. I made a circle around it. Here, I just added the description that I copy and pasted from the mark scheme, from one of the mark schemes. This is the ciliated epithelium. It's a nice diagram here, very easy to draw. In a question like this, see the question, they will ask you, describe the structural features of tissue T. You'll go to tissue T. This is what we just did. If you go to the mark scheme, okay, they will tell you accept an annotated diagram. So in this question, you can start actually by a small diagram on the side here. Okay, basement membrane, tall columnar cells, ciliated epithelium goblet cells, and label. And this will help you to answer and mention all the points, but don't do bullet points. If you want to do bullet points, because some students, they tell me that they remember better with bullet points, put the bullet point on the line itself, not like in the middle. Okay, so the 3 marks, why? Because Mariam said them. Tall columnar cells. They're tall and they're columnar. They have cilia. They have basal nuclei. Okay. And there are goblet cells in between, and they lie on a basement membrane. Now, they did not put this picture for no reason. So if you don't know how to describe a tissue, let's say they are giving you another tissue, not this one, another tissue. Look at the picture and describe the picture. Let's say they gave you a specimen from breast tissue. You never took breast tissue, and it's not in your syllabus, and you've never seen breast tissue, but they gave you a picture and they told you describe it. Don't panic. Go to the picture and describe exactly what you can see. Okay? Are you with me, boys? Now they will ask you, what is this? What is this? What is this tissue that has something inside it? Noor, can you tell me? What is X? Hello?
muscle.
Noor, it's a what?
Muscle. Muscle.
Habibi, why muscle? Well, how did you say muscle? How many times this came in a quiz and we discussed it? And you told me that you don't know how to identify. And I told you any vessel that has a shadow inside it that looks like a red blood cell, what is it? It's a capillary. Capillary. Okay? The smallest blood vessel, and it is as wide as one red blood cell. Please, when you are given any tissue specimen and you have a vessel and inside it's something like a shadow, okay, it's a red blood cell. This is a capillary. X is a capillary. Are you with me? Now, the smooth muscle, how does it look like, Noor? Faru, how does it look like? Your exam is tomorrow, in case you did not notice. Can you all
Smooth
pay
muscle.
attention with me?
It's smooth. It doesn't have any bumps or any.
Yes. Yes, please. Smooth muscle. It's a spindle-shaped cell with a central oval nuclei. Finished. That's it. And they called it smooth because it looks smooth under the microscope. Ciliated epithelium. Tall columnar cells with microscopic cilia and goblet cells in between, and basal nuclei. Basal nuclei means the nuclei is towards the base, and they lie on a basement membrane. Any layer of cells, whether ciliated epithelium, cuboidal epithelium, they always lie on a basement membrane. Good. Now, the cartilage. The cartilage always looks like a thick band under the microscope, and we have cells that are found within spaces. These cells can sometimes be singular or in pairs. I sent you all that, and I told you these are additions. Okay? Now, these spaces are called lacuna, and these cells are called chondrocytes. And the thick ground tissue around them is called thick ground tissue. That's why it looks very heavy under the microscope, like a heavy band. I sent you all that several days ago, and I keep on telling you, look at the things, look at the modification. You don't. You don't attend the online. Are you with me? Look here. I sent you here the description of the ciliated epithelium, the description of the smooth muscle, very easy, spindle-shaped cells that are smooth with a central oval nuclei, and the cartilage. The cartilage, it looks like this under the microscope. And we did see it in the lab more than once. Actually, we saw it in the last session. It is the thick band. It's thick, no blood vessels, no nerves. You have cells that are found inside spaces called lacuna. And I described it here because this will make you lose a mark or two, and we don't want that. Are you with me? Okay. Here, see, they're asking you to identify structure X. X is a capillary. How did I know? It is here. There's something inside it. This is the red blood cell, and it is as wide as a red blood cell. And of course, you know the features of a capillary. It is one cell thick, it is porous, it is as wide as a red blood cell for exchange of materials, so gases can diffuse in and out. The red blood cell is here. Okay? You're all silent, but are you all with me? Is everything clear?
Yes.
Yes.
I hope so. Describe the structure of a cellulose molecule and a cellulose microfibril. Now, this was a good chance to revise cellulose. Who will remind me? It's made up of what?
Beta glucose.
Yes. Now, of course, this question is an ideal question for an annotated diagram. You can draw the beta glucose. Remind me, how do I draw it? I forgot.
It's a hexagon.
Yeah. Okay. 1, 2, 3, 4, 5, 6.
There is a
Now,
O.
every beta glucose molecule, are they upright or rotated or what? Remind me.
One is upright and then the next one is rotated.
Yes. Now, the upright one, I'm sure you have no problems with it. Let me do it quickly. It's the inverted one that students always do mistakes. Beta, hydrogen below, OH is above. I'll teach you a way to remember the inverted. Again, I'm going to do a hexagon. 1, 2, 3, 4, 5, 6. But where is the oxygen now that it is inverted?
On the bottom.
At the bottom here, oxygen. Okay, and we will count the carbon in an anti-clockwise direction. 1, 2, 3, 4, 5, and the 6 is out, CH2OH. Now we want to create a beta 1 to 4 glycosidic bond. Okay, so here, let's go to 4. So the OH in 4 will be up or down so that we can create the bond.
Up.
Very good. It will be up so that we can create the bond. And then alternate the OH from there. So the 4 will be up, 3 will be down, the 2 will be up. Okay, and the 1 will be down. We will alternate. And you don't need to mention or write the hydrogen, and it will be correct. Because we want to create a beta 1 to 4 glycosidic bond. The OH will take an H, condensation reaction, and water goes out. And the bond will be here carbon 1. And then I have oxygen and carbon 4. Okay? So you don't get confused. You will go to carbon 4 and you will tell yourself that the OH has to be up there so that we can create the bond. And from there, the OH always alternates up, down, up, down. Clear? Now remind me, we did not finish. The cellulose molecules are branched or parallel?
Parallel.
Yes, they're parallel. And how are they held together?
By hydrogen bonds.
Very good. Okay. And this will make them strong. And what are microfibrils?
When 70 cellulose molecules joined together, they form a microfibril.
Yes, many chains of cellulose molecules will form a microfibril, and many of our microfibril will form
Fiber.
fibers that run in so many different directions. That is why the cellulose is strong, etc., etc. Okay, we answered this question in this exam. This is stomata that is depressed, and we are sunken. Now let's remember together the features of an xerophyte. I don't think you were there, Noor, when we answered this exam. What is an xerophyte?
The plants that live in a desert,
Yes, and how are they adapted to live in the desert?
they have a waxy cuticle. They have
Yes,
sunken
this is
stomata.
yes, sunken stomata, and they have microscopic hairs here. What are they called?
Um, I remember something. Uh,
Tri? Farouk, do you remember?
trichome, yes, trichome.
Yes, trichoma. Now, why does the trichoma help reduce water loss?
They trap water vapor, so that
Very
they
good.
prevent water loss.
Yes, they trap water vapor. Water vapor will be saturated here in this part. And please discuss or answer this question in terms of water potential. How will this affect the water potential inside and outside the stomata, Mariam?
The water potential will decrease.
Yes, good. The difference in water potential will decrease. In other words, I have lots of water inside the plant, and I also have lots of water outside the plant in the external environment of the plant. So the water potential difference will decrease, and this is going to reduce water loss by transpiration. Okay, Noor, please go study the features of xerophytes. Suggest the advantages to the cell of enzyme pathways being located. Now, this question also is a suggest question, critical thinking, not part of the syllabus, but it was common. And I sent you also this question. Here, they're telling you that some enzymes are located on the cell membrane. Enzymes can sometimes be present on the cell membrane, on the outer cell membrane. Why is this an advantage? Suggest advantages of locating the enzymes on the cell membrane. Give me two advantages.
It makes it easier for the substrate to arrive at the enzyme and bind with it.
Yes, you are right. Anything related to making the enzyme more available to the substrate is correct. Okay, so the enzyme and the substrate are closer together and the reaction can start more efficiently or faster. What else can you say?
The active site is exposed more when
Yes,
it's on the cell surface membrane.
yes. Also, anything related to a better and a faster reaction, that the active site is closer to the substrate, it is more exposed, it's closer to the substrate, more enzyme substrate complex, better collisions. Okay. This also, I saw it more than once. Suggest why mutations may increase the risk of cancer. I added this question because I want us to revise mutations and how do they cause cancer. Noor, can you explain this question? How do mutations cause cancer?
Random is division, يعني spontaneous division by mitosis.
Okay, yes, but you jumped.
Continuous.
Can you talk to me about the genes that are mutated that will lead to cancer?
The
Farouk, are you
tumor
there?
suppressor gene. Tumor suppressor gene is
Yes,
switched
you
off.
are right. Yes, the tumor suppressor gene is switched off. And there's another gene, only another gene that we studied. Farouk, can you tell me?
The
Where is Farouk?
oncogene,
Yes, it was the oncogene is the mutated or the healthy gene?
it's the mutated gene,
Good. So what is the healthy gene called?
proto-oncogene.
Excellent. Anytime you are asked about mutation and mitosis, define mutation, change in the base sequence of the DNA, random change. And we have two genes that control cell division. This tumor suppressor gene is turned off, and the proto-oncogene, it will change into an oncogene, a mutant gene. Now the mitosis is going to be affected. And like Noor said, but Noor jumped to the last part of the answer. He said the mitosis will be uncontrollable and we will have, what about apoptosis? No apoptosis, or we have apoptosis. And what is apoptosis anyway? No apoptosis. Cells don't die. Uncontrollable mitosis, etc., etc. And we have a mass, okay? And this is cancer. Good. Now, what is this? Yes, this was a nice one. Now, referring to this figure, explain why increasing the proportion of phospholipids with this acid in a cell will increase the fluidity of the cell surface membrane. Now, look at this type of acid, fatty acid. Why does this type of acid increase the fluidity of the cell membrane? What do you see in this fatty acid tail?
Because this fatty acid tail is highly unsaturated and there is a lot of bends, which will cause
Excellent.
it to become more fluid.
Yes. Okay, now why? How? You have here kinks. This is, we do it like this sometimes. It will increase the distance between the phospholipids. And what does it do to the intermolecular forces? More intermolecular forces or less intermolecular forces?
Less.
Yes. Okay. It will like push the phospholipids far away. They will not be closely packed. And it will reduce the intermolecular forces, revise the fluidity of the cell membrane. Okay. Part of the control of the, yes, the cardiac cycle. Now let's revise together the cardiac cycle. Noor, can you tell me the steps of the cardiac cycle?
Yes.
Huh? Now here they say outline in points, but we want to remember the whole thing. We start with what?
The right atrium.
Hmm. What happened in the right atrium?
The right atrium contains يعني gets blood from the vena cava, and
Noor,
it's
Noor,
yes.
Noor. We're talking about the cardiac cycle, not the flow of blood inside the heart. How does the heart beat? Boom, boom, boom.
Uh,
Ah. Atrial systole, ventricular systole, diastole. Who will make the heart beat? Why does the heart beat?
The sinoatrial node. Sends an
Yes,
input.
we have a wave of excitation that comes out of the sinoatrial node. And this wave of excitation, what will stimulate it, or it is myogenic?
It's myogenic.
Myogenic, of course. Now, the sinoatrial node will send a wave of excitation, Noor. And this will cause what? It will lead to what, my dear? Noor, your exam is tomorrow. Noor,
yes.
the sinoatrial node will send a wave of excitation. And it will cause what to contract?
الاتريوم.
Mariam, can you help him? Yes, yes, yes. The atria, both atria will contract. Atrial systole. Now, when the atria contracts, what will contract next and why?
الفنتريكل.
Okay, immediately, or there will be a small delay?
لا في small delay.
Excellent. Who is the cause of the delay? Mariam, who is the cause of the delay?
The atrioventricular node.
Yes, there is an atrioventricular node that is responsible for a short delay. And there is something else that contributes to this delay. And I told you to act. What is this something else?
The non-conducting tissue.
Yes, non-conducting tissue. Here, I sent it to you maybe 3, 4 times. Okay, I taught in the mark scheme, so I will add it. Good. Here, there's a nice diagram showing you the sinoatrial node. Here, it's in the wall of the right atrium, and the muscular wall of the right atrium, atrioventricular node considered in the septum of the heart. And here, there is tissue that separates between the atria and the ventricles called the annulus fibrosus. You don't have to know its name. You can simply describe it as non-conducting fibrous tissue that will also contribute to the delay in the sending of the wave of excitation to the ventricles. Non-conduction tissue that also contributes to the delay of the wave of excitation from reaching the ventricles quickly. Okay? And this is in the mark scheme. You don't believe me? Here, it's right there. Where is the diagram? There's a diagram here. Where did it go? 1 minute, I guess. Here's the diagram. See? Oh, it's right here. See? And this is not the first time I see it. There is a ring of non-conducting fibrous tissue that prevents the impulse from the sinoatrial node directly passing to the ventricles. Okay, honestly, they tell you write one from. So if you write the atrioventricular node only, you will still get the grade. But it's always better to be safe than sorry. Okay, so the atrioventricular node will delay the impulse from the sinoatrial node, and the wave of excitation or the wave of conduction will then go through the atrioventricular node and the non-conducting tissue. Outline how this control is received. They mentioned the atrioventricular node 2 times. Then it will pass to the bundle of His and Purkinje tissue. And we explained it in detail. Those Purkinje tissue, they divide into 2 so that they take the wave of excitation and they result in contraction of the ventricles from the apex of the ventricle upwards. Clear? So in order, sinoatrial node, atrioventricular node, and you can add to the atrioventricular node plus the annulus fibrosus or the non-conducting tissue between the atria and the ventricles. So the atrioventricular node and this non-conducting tissue, they both contribute to the delay. There's a delay, and then bundle of His, Purkinje fibers, apex of ventricle, and then the wave will move upwards from the apex upwards. Repeat. And the cycle keeps on repeating so that you remain alive. Okay, clear? There's a question that there's an exam that we answered together. Where is it? Where is it? Not here. We answered together here. Look. I want to show you that this question came at least 2 times in the past 2 years. Look at this. What delays the wave of depolarization between the atria and the ventricles? Look at the answer. The atrioventricular node and the non-conducting tissue between the atria and the ventricles. So you have to add it with the atrioventricular node. Am I clear?
Yes,
Hello?
miss.
I said it a very long time ago, maybe a week back. Okay, we were answering an exam together, and I noticed it in this exam, and I noticed it in another recent exam. So we cannot ignore things, even if it is just as small as this. Okay, I hope it's clear. Did we finish? Yes, we finished. Now, of course, there are so many other things that you need to revise. So I'm going to let you go and ask me any questions. If you stopped at anything or you're answering a question that you find tricky, send it to me on WhatsApp and I'll answer you immediately.
Thank you.
Okay, can you please study with a little bit of more concentration so that you're sure of your answers? Anyone has any questions? Can you please look at the modifications I sent you? These modifications, they are a summary of 2022 and 2023 exams. Last year,
Okay.
the exams ended at 21, and okay, 2022, November and June, they came or they were published at the beginning of the year. So now we solve 2022 and 2023, and I make a summary of what they were focusing on and the additions. So you have the right to look at those stuff. They can highly appear in 2024. Okay?
Okay, thank you.
Now, best of luck, and I'm here anytime you send me any questions. See you. Bye.